So, what's your name? Where are you from? And what are you working on today?
Awesome. First of all, thank you for having me here. My name is Santosh Rananaware. Currently, I'm the co-founder and CTO of a startup called CasNx. A big problem in the market right now is there's a huge number of patients on the wait list for getting an organ. For kidney transplants, there are more than 100,000 people who are on the wait list to get a kidney. But the sad reality is that maybe only 20 to 30,000 of them will get a kidney this year. Now, despite this huge shortage, it's really sad that a lot of organs get discarded, not because they're bad organs, but because they have some sort of deformity or abnormality. What we're trying to do at CasNx is we are trying to, first of all, rehabilitate these organs that are thrown away so that instead of throwing them away, you can repair them. And secondly, what we're also trying to do is organs that are already being transplanted, can we somehow enhance them or make it better so that they fail less in the patients have better outcomes, and the organs last longer in the patients. And my background is I did my undergrad in physics. In my second year, I decided to double major in chemical engineering. And then I did my PhD in chemical engineering, focused on a research in developing gene editing tools that can change the DNA and RNA of human cells. And then currently, I'm applying the research that I did during my PhD to this startup where we're trying to repair donor organs that get discarded.
Wow. Wow. And where are you based out of today?
Oh, I'm in Florida right now. That's where I
Oh,
started.
wow.
Yeah. I did my
Yeah.
PhD at UF, University of Florida.
Right.
And then since I graduated, me, my PhD advisor, and his next door neighbor, the three of us started this startup.
Wow. So those are your co-founders?
Those are my co-founders,
So
yes.
your professor and your
Mhm.
professor's neighbor and yourself.
Yes, that's right.
So I'm assuming you're the youngest co-founder. Is that correct?
I'm the youngest one, yes, that's right. Yeah.
Gotcha. And how old are you?
I'm 31 years of age right now. Yeah.
31 years of age. Okay. Wonderful. Wonderful.
And
Well,
my
it's a pleasure to
co-founder,
meet you, sir.
yeah, of course, it's a pleasure to meet you as well.
Yes. And where did you do your undergrad?
I did my undergrad in India.
Uh-huh.
My undergrad's called BITS Pilani. It's a decent university in India. And I started off doing physics
Wow.
because I wanted to understand how the universe works. It sounds silly.
Right.
And I thought physics is the cleanest expression of that. But then along the way, I decided to also, delve into chemical engineering and biology. And that's how it's like a mixture of all the different things I've done.
So when you came out as an undergrad, did you get a job right away in physics or chemistry, or where did you land?
That's a great question. Yeah. So physics jobs are very rare and very hard to get. My first job right out of undergrad was at a pharmaceutical company. And that sort of led to how I developed my interest in biology because the pharma company that hired me, they were working on drugs for breast cancer. So they were expressing these hamster cells to make a protein that would
Mhm.
act as an antibody and specifically target breast cancer cells. And, I was like a 22, 23-year-old kid back then, and the whole
Mhm.
process really fascinated me. And so that's why when I decided to do my PhD, I decided to lean more into the biological side, and that's how I ended up into all of this gene editing research that I did later on.
Mhm. That initial job, so at that first pharmaceutical company, was the modality of the project gene editing, or was it something else?
No, they were making an antibody. It's called monoclonal antibody, and so these antibodies they specifically target, specific cells. So if you want them to target cancer cells, they will target only the cancer cells and not the healthy cells. So they are a great way of selectively destroying the bad cells while keeping the good cells alive.
Right, right. So how did you first learn and where did you first use CRISPR?
Right. I'd heard of CRISPR because back in 2012, when I started my undergrad, CRISPR paper had just came out and it was, a wildfire everywhere. It would show up in news, in interviews, on YouTube, everywhere you look at. And I had no idea that I would end up doing research in CRISPR. But then when I started my PhD, I met my PhD advisors, Dr. Pius Jain, and he was the first person to sort of give me an opportunity to work in the space.
Mhm.
And coincidentally, when I started doing my PhD, he had just become a assistant professor. So it was his first year as a professor and my first year as a grad student. And so we sort of connected, and the research led from there. I was instrumental in setting up the lab, and we had great colleagues, other students around me, that we all learned how to do CRISPR together, and then eventually ended up doing a lot of research into making CRISPR systems better, more novel,
Right,
and more innovative.
right, right. So now let's talk a little bit about your startup. It's CasNx, right? That's how you say it, CasNx?
Yes,
All right, wonderful. So, and this is something I nailed David on our last interview.
right.
So, for someone hearing about CasNx for the very first time,
Hmm.
so what problem are you trying to solve? And how does the technology work at a high level?
Yes. First of all, the name CasNx is derived from CRISPR-Cas systems. So Cas are the proteins that can go and do the gene editing that is crucial of the CRISPR system. And the problem we are working on is we work with organ transplants. And so a big problem in the market right now is there's a huge number of patients on the wait list for getting an organ. For example, for kidney transplants, there are more than 100,000 people who are on the wait list to get a kidney. But the sad reality is that maybe only 20 to 30,000 of them will get a kidney this year because there's a huge discrimination between the number of organs that are available and the number of organs that are needed. Now, despite this huge shortage, it's really sad that a lot of organs get discarded, not because they're bad organs, but because they have some sort of deformity or abnormality. For example, organs can have viral infections, and so of course it's not safe to transplant them, so these organs get thrown away. And so what we're trying to do at CasNx is we are trying to first of all rehabilitate these organs that are thrown away so that instead of throwing them away, you can repair them and use them for transplants. So we can increase the number of organs that are available. And secondly, what we're also trying to do is organs that are already being transplanted, can we somehow enhance them or make it better so that they fail less in the patients have better outcomes, and the organs last longer in the patients. So those are sort of the twofold approaches that we're applying our gene editing expertise to for transplant medicine.
That's incredible. And how did this idea come about? What's like the story there? That's so unique. That's very different than what you hear usually.
Yeah, yeah, that's a great question. And as I said, I have two co-founders, my PhD advisor and his next door neighbor. So what I haven't told you so far is my advisor's neighbor is a transplant surgeon, and so he's part of the number one transplant team in the US in terms of kidney and liver transplant outcomes. And so he sees these problems on a day-to-day basis where his patients, he would transplant an organ, and weeks later or months later, patients would come back because of viral infections or because the organ's not behaving properly. It's rejecting, the body's rejecting the organ. And also he's involved in decisions whether an organ is good or bad, whether we have to throw an organ away. So he had seen all of these problems firsthand. And then, my PhD advisor and our lab has been working on gene editing systems. And so the story is that when their daughters were playing in a park, they were talking about what they do at work. And so the transplant surgeon was saying, "Oh, we have, all of these organs that we throw away. Wish there was a way to save them or remove any viruses from them." And my PhD advisor, Dr. Jain, he was like, "Oh, we actually have tools in our lab that can be added to the organ that, go in and chop off all viral DNA or RNA and make the organ virus free." So that's how we first started this company. Like, we can remove viruses from the organs. And then
Right.
eventually we realized that we can actually do not just viral removal, but also a lot more, like making the organ hypoimmunogenic so it doesn't get rejected by the patient's body, so on and so forth. And the way
Wow.
I fit in is that I was one of the first students to graduate from my professor's lab, and the timing worked out where they were having this conversation, and I was about to graduate, trying to find a job, and they of course needed someone to run this company full time because they have their obviously the professor job and surgeon job.
Right,
So
right.
they roped me in, and here we are. It's been an incredible ride. We started this company 2 years ago in 2024.
Wow.
July of 2024 is when I graduated. And it's
Wow.
been an amazing experience for me personally as well. The whole journey has been incredible.
Yeah, tell me more about that personal journey. I mean, you graduated and then you started pretty much right away, right? Same year?
Yeah, same year. The day I graduated, the very next day I started working
Yeah.
on the startup with my two co-founders. And
Uh,
it's one thing to do something in the lab, and if your science really good working in the lab, we
right.
realize there are many challenges when it comes to translating it into the clinic.
Right, right. And also tell me a little bit more about that because in theory, the science behind it is incredible, but is it still theoretical or is it proven? Is it mainstream to do this, to edit the organs?
So it's not just theory anymore. It is, but it's not yet mainstream as well.
Right,
As far as we know, we are the first company that's trying to repair or gene edit these organs
wow.
outside outside the human body before they get transplanted. That being
Right.
said, there have been studies done in academia, in research papers that show that this works, but nobody's actually tried to do it at a large scale and at a commercial scale and bring it to the clinic because it's clinical trials can be super expensive and really difficult. And what I will say though is that it wasn't possible to gene edit organs before transplant throughout history. It's only in the last 5 years that something like this has been possible.
Right.
And a big reason for that is because humanity as a species, we've gotten really good at preserving organs outside the body for a large amount of time.
Okay.
So
That
just to,
that's what I was going to ask. What
yeah.
what makes it possible now that it wasn't like 5 years ago? It's the preservation.
Yeah, exactly. So to give you an example, let's say if you were in 2012 or 2013, just 10 years or 15 years ago now, I guess,
Right.
um, whenever hospitals received an organ, they would receive it on ice, and
Mhm.
they had to rush the surgery because ice, as you can imagine, is not very great at preserving these organs. You had maybe 4 hours or 5 hours before the organs start showing signs of damage. So you have to be very, very quick in extracting the organ from the donor and then transplanting it into the recipient.
Right, right.
Since the last 5 years, there have been advancements made in this technology called perfusion pumps. And this technology is super new and it's advancing at a very, very rapid pace. And what these pumps do is that they perfuse the organs with sometimes blood, sometimes other media.
Mhm.
And these pumps, you can think of them as vessels that can keep organs alive. And so, kidney pumps, for example, my transplant surgeon co founder tells me they have only started recently using it mainstream in their hospitals for about 2 or 3 years. So, before
Wow.
that, it would all be on ice. And now, a majority of their organs are on the pump. And
That's incredible.
kidney pumps, whereas ice could preserve kidneys for maybe hours, kidney pumps can do it for 24 hours to 48 hours. So, it's a significant
Wow. Yeah.
increase in the amount of time organs can stay outside, alive outside the body. And so, finally, for the first time in history, we have the time needed to repair these organs before they get transplanted.
My
That's
God, that's fascinating. And previously with like just ice, how long would an organ typically like last for before it had to be implanted?
Yeah, so that depends on the organ, right? Some organs, different organs have different times. I'm not an expert in that, but I would guess you had
Right.
maybe around 4 to 6 hours for
Wow.
on an average for a typical organ.
Right.
And pumps have increased that from maybe 4 to 6 hours to sometimes even 24 to 48 hours. And pump technology is getting better and better every day.
Mhm.
They used to have initially the version one of the pumps were hypothermic pumps. So they were cold pumps that would, flow cold media
Mhm.
through the organ to keep it alive, to mimic ice.
Right.
But then now we have normothermic pumps, and these pumps are even better. And what these pumps do is instead of flowing cold media, some of these pumps flow like warm blood. So the organ
Mm.
feels like as if it's still in the body. And,
Wow.
the amount of preservation time we get from that is incredible. And so we finally have the time to go and repair these organs.
That's incredible. You mentioned kidney a few times. Is that like the number one organ that you guys are testing with, or what organs are you doing?
We're starting out with kidneys. We've also done livers, but
Okay.
kidneys especially interesting because, first of all, my co-founder's expertise is in kidney transplants, and
Right.
secondly, kidney is the highest volume transplanted organ because you get two kidneys from every donor as opposed
Mhm.
to one heart, one liver, and then the number of people needing kidney transplants is a lot more, and so because it makes a lot of sense to start with kidney as our first, organ.
Right, right. And what's the advantage to doing the CRISPR, the gene editing, outside of the body instead of after they've already transplanted it to the patient? Is there an advantage to it? Like, why not just do it then?
Oh yeah, there are tons of advantages. We at CasNx, we think it's like the way to go about with gene editing because
Hmm.
you're working inside a patient in vivo. A lot of things can go wrong. There's safety issues. Gene editing tools notoriously have off-target effects, so it's really
Right.
risky to do it in a patient. Delivery is a big problem too because when you deliver something into a human body, our immune systems try and attack it. It thinks, it's a foreign object, and
Right. Right.
subsequently you need a lot more dosage so that the right amount of dosage can reach your target cells. When
Hmm.
you do it outside a human body, which is called ex vivo, it's a completely different ball game. Suddenly, it's
Hmm.
much easier to deliver it because you can just soak in our case, you can soak the organ with a large amount of the CRISPR editor initially and then wash it off before you transplant it into the patient. So there's effectively no CRISPR that's going into the patient, and you're just treating the organ itself. It's
Wow.
easy to deliver. It has, even if you have off-target effects, you can really characterize the organ before you transplant it. And if you have really bad case of off-target effects, you can, simply not transplant it. It's a lot safer.
Wow.
Um,
So generally, it's a lot safer for the patient. There's a lot less, I guess, complications.
lot less
Right.
complications. It's cheaper. It's easier. It just makes a lot more sense to do it. And secondly, the way transplant medicine is done today is, for example, with viral infections, the patient would or the surgeon would transplant the patient with an organ.
Right. Right.
If they have viral infections related to the organ later on, the patient gets an antiviral treatment course, which can go up to 12 weeks and costs a lot of, money and, you know, problems to the patient. Instead of all of that, we front load all the expenses, front load all the problems and, fix it beforehand. So the patient doesn't have to do anything afterwards. The patient just gets a healthy organ.
That is incredible. That is incredible. How, this is a very scientific question. How do you eliminate the rejection aspect of putting an organ into someone's body? How do you eliminate that with CRISPR? Very high level.
No, yeah, no, that's a great question. And organ rejection, honestly, we don't really understand completely how that
Right.
happens. We do
Right.
know that on the organ side, there are certain DNA or RNA triggers that the immune system recognizes and starts attacking. And so
Uh-huh.
the idea is that you apply CRISPR to the organ and you suppress all of these DNA or RNA triggers that your immune system recognizes. And
Right.
hopefully, because you suppress them, the body doesn't recognize that, it makes the organ sort of into a stealth mode where the body doesn't recognize it's a foreign organ and try and attack it. That being said, this is this part is still theoretical. So we are not sure how many RNAs we need to suppress to get really good rejection,
Hmm.
rejection avoidance per se.
Hmm.
And this is something that still needs we are experimenting with basically. But
Right.
in
Right.
theory, it has been shown that if you suppress certain DNA or RNA triggers, you can at least reduce the rejection episodes.
I mean, that in itself would be a game changer.
Yes.
That would be absolutely huge. My God, that's incredible. It's very exciting to think about. So you recently worked on research involving DNA-guided CRISPR. In simple terms, what did you discover from that research, and I guess why could it be important?
Yeah, that's another great question. And so I'll start off by saying that CRISPR, people hear of this as a gene editing tool, but it's really made up of two different components. There's an enzyme called the Cas enzyme that can go and cut genes. And
Right.
then there's a piece of RNA, it's called the guide RNA, that tells
Mhm.
this Cas enzyme where to go and cut. And so you can design the guide RNA to target different genes, and then the Cas can go and cut these different genes. The problem is, so this is how it happens in nature. These CRISPR-Cas enzymes are found in bacteria, and
Mhm.
bacteria use it as a defense system against viruses. And throughout nature, the Cas enzyme is a protein, and the guide piece is a piece of RNA. Now, the problem with RNA is that it's very finicky. If you have to synthetically make a piece of RNA, it's much more expensive, it's much more difficult, and it's a lot less stable than DNA molecules. DNA or RNA are very similar chemically, as well as they just have like one element that's different. DNA is a deoxynucleic acid molecule, so there's just one oxygen atom that's different between a DNA and RNA. But because of that, DNA molecules are a lot more stable. And so what we discovered in our research is that even though naturally all CRISPR-Cas systems are RNA guided, we were able to use a DNA mimic of the guide RNA to sort of guide now Cas enzymes with DNA molecules instead of RNA molecules. And the reason why this is so important is because suddenly you have a molecule that's tenfold easier to synthesize, tenfold cheaper to synthesize, and also like lasts longer uh, inside for our applications, especially for our applications where we have to deliver these CRISPR and guide into like organ tissue, which
Hmm.
notoriously has a very harsh environment. The DNA molecules survive this harsher environment a lot more stably and a lot longer than RNA molecules. That's the advantage.
Do we know why that is? Why the DNA survives more than the RNA?
Yeah, because in the cells there are a lot of things that attack RNA, but very less things that attack a DNA. And
Uh-huh.
so DNA molecules in general in nature, they are a lot stabler than RNA molecules. Also,
Right.
naturally occurring DNA is mostly double-stranded DNA, whereas naturally occurring RNA is, for the most part, single-stranded RNA. And so
Hmm.
this double-stranded nature of DNA makes it a lot more stable as well. And it's
Uh, Yeah. I'm wondering why they even started using RNA instead of DNA. Do you know the background on that or no?
yeah, of course. I mean, that's how CRISPR systems are discovered in nature, right? So in nature, it's all RNA. It's
Uh-huh.
RNA Cas enzymes. You don't have DNA guided CRISPR in nature. And so for a lot of time, we just didn't know how to make these CRISPR systems work with DNA. People
Hmm.
tried, but you know, there was no success. And then in our lab, we had this insight of, I mean, this is kind of like an accidental discovery where we added like a RNA like hairpin to the DNA piece and suddenly started working like a guide. Um,
It's incredible.
and so to answer your question simply, it's like RNA guided CRISPR is found everywhere in nature. There's no DNA guided CRISPR. But in our lab, due to some accidental discoveries, we were able to make DNA guides work with CRISPR as well.
That's fascinating. You made an interesting point there. You said RNA-guided CRISPR is everywhere in nature. Tell me more about that because I don't know anything about that. How do we see it in nature?
Yeah, so what happens in nature is that these CRISPR-Cas systems they are found in bacteria, and bacteria get attacked by viruses. They are called bacteriophages. So these are viruses that go and inject their genetic material into these bacterial cells. And so to counter that, what bacteria have come up with are these CRISPR systems that are made up of a piece of DNA or RNA chopping enzymes and a piece of RNA. Now,
Hmm.
why they evolved RNA and not DNA is a different question. We don't really know, but the fact is they evolved to be an enzyme, a protein that is guided by an RNA that can go and chop off these viral invaders whenever they attack, a
Hmm.
bacterial cell. And so because it evolved to have RNA, that's how we discovered it. That's how we've been applying it. And then, in the lab, we were able to make DNA work as a guide as well. So
Wow, wow. And when did you do this specific research? When was this done?
this was done towards the end of my doctoral program. And so I'd say my PhD lasted a total of 5 years. So this was done during the third year and fourth year. So the fourth year and the fifth year, the last 2 years of my research.
Right. And this was done at the University of Florida, you said, right?
That is correct. Yes.
Right. That's incredible. That's incredible. And you did that research under the guidance of your current co-founder, correct? Or is that separate?
No, under the guidance of my current co-founder. And so
Wow.
when we were doing this research, we realized that this can have immense potential, but we
Right.
just had a tool and we didn't know what problem to apply to.
Right.
And so when my advisor had a new neighbor who happened to be a transplant surgeon, it sort of worked out where, oh, we have this cool technology and now we have a problem, a real problem that a lot of people are suffering from that we can apply to and, have an impact that way.
That's incredible. That's incredible. If I can go off on a tangent here, I want to hear, more about the business aspect too. Are you guys fundraising? Are
Hmm.
all of you have some sort of business acumen, or one of your co-founders more focused on that? What does that look like? What's the business side look like?
Yeah, we are actually fundraising. As startups, I think we are always fundraising, especially for deep tech startups like us, because you need a lot of capital up front to prove the technology is working and it's safe to use in humans. And so yeah, we are raising a seed round, if you want to just put it out there. We are all very academic background. I did my PhD. My PhD advisor has been completely academic background. He was a postdoc at MIT before. My transplant surgeon co-founder is obviously a surgeon. So
Mhm.
none of us have business expertise per se, but it's something we are developing as we work on this startup more and more. Uh,
Mhm.
so you're forced to adapt and learn. And we do have really great mentors, and we've been fortunate that some of the people around us, they're really interested in our mission and what we're trying to do. And even though we might not have that expertise, we have access to people who have business expertise and have been guiding us throughout these journeys.
Right.
And
Right.
yeah,
Yeah, it's more science focused, but I mean, it's beautiful. There's a beauty to it because it's really just some deep, intelligent stuff there. Tell me about, pitching. Have you been involved with the pitches, or is it more your co-founders, or what does that look like?
all of us pitch at various points of the startup, but pitching, I think I've been doing most of the pitching for the company since I'm the full-time worker at the company.
Right.
Uh, in terms of pitching, our pitch has evolved significantly over the last 2 years. I can tell you that, as we've learned more and
I
more.
bet.
It, I tell you, the biggest thing I've learned is that initially, when we started out with the startup, my pitch would be super science focused and super into what the technology is, how cool the technology is, and
Right.
how great of an advance it is, because that was interesting to me. But
Right.
then over the years, I realized that nobody cares what technology you're using. What people more care about is what problem are you solving? How will this problem affect and change the world? And so it's more about the storytelling aspect of it rather than the technology, because people just trust us with the technology, because a lot of people don't have the expertise that is needed to really
Right.
understand what makes DNA guided CRISPR cooler than RNA guided CRISPR, for example. But
Right.
everybody can relate to the idea that okay, you can repair these organs before transplants and somehow make better outcomes and make patients' lives better. So
Right.
that's
How has that been for you, taking this very densely scientific topic and kind of breaking it down into something a little bit more simple? How is that mindset switch for you? How did you get there?
yeah, over time in chunks. So I can tell you very confidently that the first 20 pitches I did, they were all very terrible. But through each pitch, I got a little bit of feedback. Okay, I didn't understand this part. I didn't understand this part. And then we just
Right.
kept refining and refining and refining to a point where eventually I think we have a great pitch now. And, it's just over time, your story evolves to be something coherent,
Right,
something
right.
easy to understand. Yeah.
So was it more so feedback from either judges or VCs or investors or angels, or was it just kind of a mindset switch of, this doesn't seem to be working. I need to switch it up a little bit.
A bit of everything. So, in the last 2 years of our journey, we have been part of different accelerators, particularly Nucleate Accelerator, which is for anyone who's doing biotech. I really highly recommend them to join Nucleate. And then also, I was recently part of Draper University, and I want to give them a shout out as
Nice.
well.
Is that how
That's
you met David?
that's how I met David as well. And,
Wonderful.
people at DU, the mentors we met, as well as the mentors I met at Nucleate, they were incremental, or they're, I mean, super critical, I meant, to help me refine the story of the startup. And I've also learned a lot from just my friends, pitching to my friends, and also pitching to VCs. Although VCs, I'd say, like, from in my experience, I pitch to a VC and they always say good things about your company. They're never going to say bad things about your company. So it's sometimes hard to get like true feedback from them as to what they're thinking. But then everybody else around us, people who are, interested in the company or interested in us, they've been giving us feedbacks, and that's how
Right,
we
right.
progress.
When you're pitching to a VC, so we'll go VC specific right now. Are you guys targeting like life science VCs, or is this very general VCs? Is it San Francisco VCs or is it Cambridge VCs? Like, what does that look like for you guys?
I mean, we are just brute forcing our way to every VC who would give us their time, basically.
Right, right,
But we do have a ranked list, so we do go with life science VCs first. We try to think of people who might be interested in kidney specific problems or gene editing, who are interested in gene editing.
Mhm.
The biggest tell is if they've invested in similar companies before. So what we actually do is we look at other companies who are doing similar stuff to us, maybe not necessarily related to organ transplants, but maybe they're also applying gene editing for some other application, and we see who's funded them, and that's how
Mhm.
we create our list, and then we sometimes do cold outreach, although that rarely works. But we try to connect with these other startups who are doing similarly cool stuff as us, and then try to get introductions to their investors.
Right.
A warm intro always goes a longer way than cold
Of
outreaches.
course. Of course. Yeah. Tell me about Draper University. Were you in the same cohort as David?
Yes, yes, I was in the same cohort as David. It actually ended
That's
fairly
very
recently.
cool.
So we
Right.
were part of the spring cohort that lasted from end of April to sort of end of May. It was a 5 week course, and it's a great program. Like I not, don't have enough good words to say about it. Yeah.
Yeah. Did you do that same act? David was telling me about this ridiculous activity where they had you guys sell condoms to strangers.
We had to sell condoms to strangers, and then they took us to a 5 day military style boot camp where we were underfed and we were just made really miserable. And somehow it's still the best part of everybody's experience of the whole program. Part of it involved, by the way, was I don't know if David told you this, but we had to jump into an ice bath and pitch our companies to a billionaire for
Yeah,
about
Tim Draper, right?
Tim Draper. Yeah, yeah,
Yeah,
exactly.
yeah. I saw that on LinkedIn actually. I didn't know you were involved. That's so cool.
Yeah, yeah.
How was that experience for you? Were you able to actually get your pitch out or were you too cold?
So, funnily enough, I had heard about this ice bath bridge even before I went to Survival Week because there are, accounts of people who've done it in the past saying,
Right.
and I was really scared of it because I'm super scared of ice and super scared of being cold. Um, but then when I actually jumped into that ice bath, funnily enough, it wasn't as bad as I'd imagined it to be. And so I
Right.
was fine for the most part. That was probably the easiest part of my whole, 5 days there. Uh,
That's
it's the
incredible.
activity I was most worried about, interestingly. Yeah.
Do you remember? Did Tim give you any feedback that kind of stuck with you, or do you remember anything from it? What did he say to you?
Tim gave me a lot of feedback throughout the program. For all of us had, I think, a really amazing, a lot of time, one-on-one time with him to,
Right.
really pick his brain and get ideas from him. I
Right.
don't remember what specific feedback he gave me in that ice bath, but overall, he gave me really good points, and I'm grateful for all that he's done. Yeah.
So,
Yeah.
what are some of the biggest challenges in taking something that works in the lab and eventually turning it into something that could be used clinically in a clinical setting?
Yeah, that's great. There are a lot of challenges.
Right.
Just because the science works in the lab doesn't mean it's easy to take it and just bring it to the clinic. And the key challenge is that, I mean, there are a few of those challenges, but the biggest one I think is the scale. Because when you're doing it in the lab, everything's at a small scale, and it's easy, it's cheap to iterate. But when you're doing it at a clinical setting, suddenly every experiment costs 100 times more than what it did in the lab. And so you have to be really careful and plan it out well and hope that this experiment works, which it might not before you put a lot of time and resources into it. And so every decision just gets a little bit more difficult or more crucial than in the lab. In the lab, you iterate, like you do an experiment, even if it's wrong or if you made a mistake, you can just repeat it again within a matter of days.
Right.
Uh,
So clinically, it becomes even more capital intensive, you have
Exactly.
to be more cautious. You have to have the steps lined up very carefully.
Yeah.
What is, like, theoretically, and I'm sure you've been thinking about this, what is your remedy to that? How do you solve that?
You have to really minimize human like force. There are forced errors and unforced errors, right? So you have to minimize forced errors. Unforced errors will happen no matter what you do,
Right,
and planning really carefully every single aspect of what can happen and what can go wrong is really important.
right,
Yeah.
right. Yeah, I'm sure that's something that's plagued your mind.
Hmm.
Yeah.
Yeah. Yeah.
Okay.
Yeah.
All right, so related. So as CasNx moves towards the clinic, what kinds of CROs, CDMOs, or CMOs, or regulatory partners do you think you're going to need along the way? Or have you already started, engaging with them?
No, we are a little bit too early for having partners, but
Right,
I can imagine we would need partners who can develop the enzymes and the DNA or the RNA molecules that we need. And so
right.
there are companies like IDT, Integrated DNA Technologies. They're great at manufacturing these components. So
Hmm.
basically, the raw material that we need to go and develop our drug. We've actually spoken to them already related to another project. So we have some sort of connections there already. We'll also need someone who's an expert, like consultants who are experts in guiding us through FDA regulatory approval.
Right,
Because
right.
honestly, we have some idea, but we are not experts in how to navigate that whole. Because it's also a really expensive part of the whole
Right,
process. The clinical trials itself will cost us millions of dollars before we
right.
get to the.
I was going to ask if you guys had a theoretical CMC pathway, but it sounds like it's too early, right?
No, we have. This is something we get asked a lot, and we've been thinking about it, and so.
Right.
Uh, this is what we're doing is something relatively different than what's already out there, but
Hmm.
we have some already FDA approved CRISPR drugs that are currently in the market. So we think we are going to use them as our example and sort of follow the same pathway that they did.
Right.
And luckily enough, the CRISPR drug that's already approved by the FDA, it's also an ex vivo drug. So it's called CasGevi. I don't know if you've heard of it. It's for sickle cell disease. And
Right.
so that whole treatment also happens outside the body ex vivo, which is sort of also what
Right.
we do with organs. And so we are hoping to use them as a, precedent to go on a
Right.
similar regulatory pathway.
Yeah, it's like a predicate almost. Yeah.
Predicate, yeah,
That's
exactly.
phenomenal. That's phenomenal. Um, this might be too early to ask this, but I'm just wondering, what is your and your team's thoughts? Are you looking for like a large scale manufacturer, something like a Catalent or a Lonza or a Thermo Fisher, something smaller size, or something like very niche size, like a mom and pop kind of prototyping lab? How do you feel about, those different manufacturing partners?
Honestly, we don't really care the size of our manufacturing partner as long as they can meet our specifications in a timely manner and in a
Right.
cost-effective manner and be fast. So whether it's a mom and pop shop or like a big giant like Catlin or Lonza,
Right.
doesn't really matter as long as we get all the things that we need efficiently.
Right. Right. Wonderful. Not on here, but I want to ask more about your challenges. So, from the start, where you are now, what kind of challenges have you been facing? Whether that's about the business, about the science, your own mindset. How has your journey been?
There has been a lot of learning. Um, of course, all me and my co-founders, all of us are first-time founders.
Right.
And so it was a lot of this startup lingo we had to learn, seed, pre-seed,
Yeah.
safe. Um, so sorry, that part is something that was new to us, and it's been kind of fun to explore and learn from, honestly. The other side is I'm a chemical engineer. My co-founder is a transplant surgeon. So he had to learn all of the, CRISPR and DNA, RNA stuff. I mean, he's a doctor, so he already know part of that already. But for me personally, I had to learn how to work with organs. I'd never worked with organs before.
Right.
And that was also a big learning experience. Our tech worked really well in the lab, but we had to figure out a way to deliver it inside organ tissue. Because so
Hmm.
far, the experiments that we'd done before the startup were all in like models, cell models.
Hmm.
And move from a cell model to a real human organ, there are challenges because every human organ is different. They'll have different DNA. And so we had to figure out a way to sort of normalize all of that and
Hmm.
you know, create a standard procedure that will work with different types of organs, even if they have individual differences within them. And then the scale at which we do things change too, because a whole organ is, a lot larger than what the cells we work in the lab. And so everything had to be done at a higher, larger scale. So that was also kind of challenging.
Right, right. Yeah, I imagine you guys started with animal organs, right? Before you jumped to human, or you started with human.
No,
Really?
thankfully, due to my co-founders' connections, we collaborated with the OPO, an organ procurement organization, and
Wow.
they were kind enough to donate to us all the organs that they would have otherwise thrown away because
Wow.
they get a lot of bad organs that they just have to
Right.
throw. So instead of that, with the consent of the patient, they were able to give us these donor organs for research instead, and that's what we've been using. So we got access to real human organs relatively quickly, thankfully, in our journey, which is an advantage of our startup, I think.
That's incredible. Yeah. So I imagine you have those transfusion pumps that you mentioned earlier. You have them in lab.
No, so that's the other challenge we had. Those
Ah.
pumps super expensive, right?
Yeah,
So
yeah.
what we had to do is we had to rig our own pump. So
Wow.
we basically create like a do-it-yourself pump to sort of, yeah. We also got a second-hand pump, but it doesn't really work that well. But it's really useful for show and tell. When we go to investor meetings, we go with that second-hand pump, but it doesn't work that well. So all our experiments are actually done on like the rigged pump that we rigged together, right?
That's
For
so funny. So the real one, you just use it for show.
yeah, exactly.
That's phenomenal. That's so funny. That's amazing though. That's amazing though. And when you get those organs, I think I already asked this, they're mainly kidneys, right? Or are they all sorts of organs?
Mainly kidneys. So far we've received around 10 kidneys in the lab and
Right.
two livers. So we've also worked with livers.
Jumping back to my question about CDMOs, CMOs, CROs. I know, and this is a little bit earlier, but when that time comes to work with those outside partners and vendors, how do you plan to find and evaluate and kind of identify the right ones? Is there a plan or is it premature?
I mean, just talking to a lot of these different vendors before you make a decision would help a lot. What
Right,
you're primarily looking for is, have they worked with someone similar to us in the past? So
right,
do they have a lot of experience working with startups, or do they have a lot of experience working with other companies who have same sort of requirements as us?
right.
And so it would go a long way in our decision making, I think.
I see, I see. Okay. So mainly like, do you have experience working on a very similar type of product? That's the
Mhm.
main thing. Okay.
Yeah.
So last question for you. We would love to check back with you in a few months to a year, see how
Yeah.
things have progressed. Putting you on the
Right.
spot right now a little bit, where do you hope that CasNx will be by then?
Oh, that's another great question. Hopefully, uh, so to give you context of where we are right now, we have the CRISPR technology and we've shown it works in organ tissue. So whenever we get an organ, we chop it up into thin slices and then we do our gene editing on those thin slices because it's cheaper that way.
Mhm.
And what we're trying to do is we are fundraising to do our first whole organ experiment, where you take a whole organ and then do gene editing on all the trillions of cells that the organ has, which we are super excited about and which
Right?
nobody has done yet. So hopefully, if you when you check back on me, we would have done that experiment, have had similar results, and
Right.
progress from there.
Right. All right. We're hoping you the best. We're hoping that you raise the capital, you get that done, and promising results.